like to thank the organizers for inviting me to come and speak today and share some of our work that we've been doing in the field of mammalian synthetic biology. And so today I'll sort of review what our approach has been and the types of tools that we've been focused on developing, and touch a little bit at the end to some of the applications that we've been looking at as well. Okay. Just 1 second. Okay. All right. Okay, so just as a way of introduction, my lab has sort of worked in different areas of synthetic biology. In particular, we certainly work on the development of different foundational tools that will enable the programming of different types of cellular behaviors. And I'll focus on one class of tools today, the input-output tools, and how they can be implemented within the types of circuits that we develop in mammalian cells. These tools sort of feed into scalable biological computation, so different types of larger biological information processing circuits that we might implement into systems. And then within the application space, we sort of have been looking at two areas, and in particular today, how these types of input-output tools and biological computation circuits can enable us to do new things in the area of therapeutics. And so we've looked a lot at these input-output tools and how to build them specifically out of RNA as a substrate. And so I'm going to just briefly review sort of the approaches that people in the field have taken to the development of these types of RNA-based gene control devices. And so the basic idea is that we can identify RNA molecules or sequences of RNA that exhibit these different component functions of sensing, actuation, and information transmission. These sequences of RNA can actually be derived from a number of different sources. Oftentimes, we'll harvest them from natural systems. So there are many, many examples, and the list is just sort of growing, of RNA molecules that can actuate gene expression, and also RNA that can act as a sensing molecule for different types of proteins, small molecules, and RNAs within the cell. There's also a lot of laboratory evolution that goes on in developing these molecules when you don't have natural components to go off of. And because the function of RNA is largely dictated by its secondary structure, and we have folding algorithms to predict that, there's a lot of computational design that goes into the development of new types of RNA sequences. And then finally, these components get integrated and pieced together to build these types of input-output tools. And researchers have shown that you can develop these kind of sensing actuation RNA molecules that can respond to a wide variety of inputs, including temperature, other RNA molecules, as Jim was sort of mentioning, and small molecule and protein responsive devices. And my laboratory in particular has looked at the bottom corner of this, and that's the examples that I'll focus on today. So why is this interesting, and why might we want to think about sort of programming with RNA? I think there's been many examples now in the field, and so people are more used to thinking about this, but this was a question that came up quite a bit early on when we started this work. And one of the reasons that this can be a very powerful platform is the ability to develop novel sensors de novo out of RNA molecules. And so there's these processes that are sort of referred to as CELEX or in vitro selection, where essentially you can start with a very large pool of random RNA molecules on the order of about 10 to the 13. You can take your target of interest, whether it's a protein biomarker or a small molecule hormone that you would like to develop a sensor to. You incubate them, you partition the very small number of bound sequences from this large library, and then amplify and sort of go through this process iteratively. And so people have shown that with this process, you can develop de novo sequences of RNA that can respond to a wide variety of different types of ligands, including proteins and peptides, small molecules, and even lipids and metal ions. And so the important part is that we have technologies, or at least procedures, that allow us to go into the laboratory and basically do this type of evolution or screening for new sequences of RNA that will bind a target of interest for us in an application space. And this becomes very important when we think about mammalian synthetic biology, because if we're talking about therapeutics, there's sort of a limited space of input molecules or molecular signals in the environment that we would like the cells to be able to respond to. We're talking about things like pharmaceutical drugs that are FDA approved, or protein biomarkers, or markers of disease and infection. And so, we need to have basically sensors that can actually allow us to access that type of information very specifically. And the standard types of small molecules that we might use, like IPTG or ATC, et cetera, are not really going to work when we talk about going into a human patient. So my laboratory has done a lot of work basically in developing these RNA-based gene control devices, where again the basic idea is linking RNA sensing function to RNA actuation function. And just as a way of overview, and then we'll dive into a couple of examples, we have developed systems that work through ribozyme-based cleavage. We've also developed sort of RNAi-based systems that either work through shRNA-mediated silencing or microRNA-mediated silencing. And then even more recently, we've started to work with systems that actually modulate alternative splicing. And as we'll sort of touch upon in my talk, I won't go through all of these examples, but I'll focus on a couple. The reason that you actually want to link into these different types of regulatory mechanisms is that they will be more or less appropriate for particular targets that you go after. So the RNAi-based targets could be very useful when you're looking to target endogenous genes, so not transgenes that you put into the cell. The ribozyme-based platform can be very useful when you're targeting transgenes, the control of transgenes. And alternative splicing becomes very interesting when you're thinking not just a sort of gene regulation, but actually of protein diversity. So if you have proteins that sort of have these modular domains, like zinc fingers or talons, and you want to begin to sort of dynamically reprogram their activities and where they might sort of function and hit different parts of DNA, you can actually sort of actively begin to reprogram them with this type of technology. Okay, so let's first go into the example of these ribozyme-based control devices. So this slide is just giving you sort of an overview of how we design them and then how they're actually designed to work. And so again, the idea is that we would identify sequences of RNA that have the sensing function we desire, have the actuation function that we desire, which here is a self-cleaving ribozyme or self-cleaving piece of RNA, and then a transmitter element, which essentially links the two. And then ultimately, these get integrated into a single RNA molecule, as you see here. And in this case, it's linked to the gene of interest, which is why we say it's useful for regulating transgenes. And the sequence of RNA is essentially designed to adopt two primary conformations, although more likely it's adopting more than just the two that we illustrate here. And what you can see is that in this state, the ribozyme would be active and you would get cleavage, and therefore your transgene expression would be turned off. In this state here, you can see the structure has been altered. It's binding to its ligand molecule. The ribozyme is inactive, and gene expression would be on. So that's essentially how we design these molecules. Now, the interesting thing about this platform in particular is that because we're using a ribozyme, it doesn't depend on any cell-specific machinery, right? So, for example, if you were designing this with a microRNA, you would want to use it within a host cell that actually has the RNAi sort of processing pathway. But in this case, a ribozyme doesn't require any sort of host-specific machinery. And so in addition to sort of this modular design where you can plug and play in different components here to tailor your system, you could also imagine that this particular design allows it to be portable across different types of host. And in particular, we had done a lot of our early development in a microorganism, yeast. And what we've shown more recently is we can take the same sort of RNA design and not have to redesign it at all and show that it directly ports over into mammalian cells when, of course, you replace the promoters with promoters and terminators that will work within mammalian cells. But the RNA device itself does not have to be redesigned. And so I just want to go over some examples of that and sort of show you, and then talk about how that might be useful. So again, if we come back to our ribozyme design, we would have our RNA device, which is here. When the ribozyme is active, it cleaves the transcript, and that will inactivate the transcript, so gene expression would be turned off. When your ligand molecule is present, that will sort of shift the ribozyme to go into this alternative conformation where the ribozyme is inactive and gene expression is on. And so we could put this type of construct into mammalian cells, and if we use it to regulate GFP, and we basically do a transient transfection, okay? So this is sort of a less neat experiment than if we were actually doing a chromosomal integration and we look at the mean fluorescent intensities of the cell. In our first design, you can see that there is some response to the ligand molecule, which here is a purine alkaloid, but there's a lot of leakiness in the system, right? And so what we've also shown is that if we specifically go to the transmitter component of the switch, and we basically start to make small changes to the sequence of that transmitter, that affects basically the energetics, both the kinetics and the thermodynamics of which it adopts those two different conformations that I showed you, sort of this conformation and this conformation, and that ultimately impacts the switch dynamics that you see here. So you can see that there's two additional iterations that have just modified the transmitter beyond the sort of initial design. And you see that we're able to modify the basal level, the stringency of the switch, and see increased switching. And then what we can also show is that we have a modular platform in which we can swap out the aptamer for a purine alkaloid theophylline and put in the aptamer for tetracycline, make no other design changes to the system, and get, in this case, 2 switches with 2 different transmitters that respond to tetracycline. So this is just to show that we have the capability of tuning the system, and we also have the capability of doing a direct aptamer replacement to tailor it to different types of inputs. Now, the other thing that we can do with the transmitter is we can actually alter the transmitter in a more significant way so that we alter the kind of computation that's encoded within the switch. So in the previous example, the sort of non-ligand bind state was the ribozyme active state. In this example, the transmitter has been redesigned so that in the non-ligand bound form, the ribozyme is inactive. You get gene expression in this state. When your input is present, now that switches the conformation to the ribozyme active form, and your gene expression should be turned off. And now, instead of building an off switch, you can see that we're able to build an on switch, again, just by focusing in on modulating the sequence of that transmitter. OK, so another thing that we can do to sort of tune the stringency of the system beyond transmitter tuning is actually doing multi-copy integration, right? And so in this case, what we're proposing to do is that for a given ribozyme switch that's in the downstream part of the gene, there's a certain probability with which it will be present in the cleaved conformation, okay? And that's going to basically set the basal level that you see here. As you add additional switches into this construct, essentially you just increase the chance that any one of those switches will be in the ribozyme cleaved conformation, and all you need is one of them to be in the ribozyme cleaved conformation to inactivate the transcript. So you're basically increasing the probability by which you would expect a transcript to be cleaved and inactivated. And what we can show is that if we go from a single copy to 2 to 3 copies, we see a systematic decrease, okay, in the basal level of the switch, as you see here, and the entire system maintains its switching activity, as you see here. If we actually go and do this for a series of different devices that have different transmitters and look at single and triple copies, and then we look at the measured levels versus the computationally predicted levels, you can see that there's a very good fit. And what we mean by computationally predicted levels is essentially just that you would expect this effect to be multiplicative, so that if you take the basal level that you sort of measured from a single copy and then just sort of started to multiply it for the 2 and 3 copies, you can see that the actual experimental values match very closely, so that the system is sort of behaving in a modular fashion, and each of these switches is acting independently as we would expect. The other thing that's important to note about these systems is that they're titratable, right? So, and this was sort of mentioned in the previous talk, these are not sort of this is not a digital switch where you just sort of have an on-off, as we're showing here at the sort of low and high level, but that if you actually titrate the system across the concentration of theophylline, in this case, you see that you get a titrated response of the, here we're showing the single copy and the triple copy in red and purple. And so you can see that you're titrating the response across the entire range that you see here. And what you also note is that, in particular for the switches with the lower basal levels or the greater stringency, is that even at the high level of your ligand, you're not actually saturating the response, right? So that if you could add more ligand, in this case, at this particular concentration, this ligand becomes toxic to the cells, so we don't go above it, you could actually push it to a higher on state. Okay, and so what we were also able to show with this system is that, and what I had sort of indicated before, is that we had done a lot of our initial development of this RNA device in yeast, in part because yeast is a simpler organism to work with, and we can do more sort of prototyping of different designs in yeast. And so what we were able to show is that when we took the systems that we developed in yeast and then moved them into 3 different human cell lines, so HEK, HeLa, and U2OS, if we then just sort of compared for each point as sort of an individual switch, the basal level, so the expression level in the absence of ligand, and the induced level in the presence of ligand, you could see that there was pretty good correlation between what we were measuring in mammalian cells and the human cells and what we were measuring in yeast. So that there's sort of a direct translation when we characterize something in yeast and then move it over into human cells, we can sort of predict, right, what behavior it would essentially have when we move it into the human cells. So why is this interesting from sort of a synthetic biology and development point of view? So, you know, I think has been so probably sort of mentioned throughout the talks, right? when we do this type of design, there's a lot of iteration, right? And there's a lot of sort of iteration and redesign to kind of get the behavior that we would like to see. When we work with human cells, and probably a lot of what you've heard today are sort of immortalized cell lines, right? So model cell lines like HEK and HeLa. It's even more complex when you start to work with primary cell lines, right? Which is actually what you'd want to be doing when you go into particular applications. It's very hard to sort of screen a lot of different designs, right? It's just more complex to work with the cell lines. And so if you can develop a rapid prototype platform where you do a lot of your initial design and optimization within simpler systems, either in vitro or in a simpler organism like yeast, and you can sort of predict how that will behave when you port it over into your application cell line, you can do a lot with more rapid design. And so this is kind of the pipeline that we've been setting up in the lab right now, where essentially in vitro, the ribozymes, of course, also exhibit their cleavage activity in vitro. They don't have to be in a cell to do that. And what that means is that within a test tube or outside of a cell, you can actually set up these cleavage reactions that sort of link binding to cleavage, and then you can basically set up these type of enrichment reactions where you either recover the cleaved or uncleaved form of the ribozyme as a function of the ligand that you put in. The reason why this is interesting is that that allows you to really get to the very large libraries, the sort of 10 to the 13, 10 to the 14 libraries that you really need if you're going to go in and develop sensors to new types of approved pharmaceutical drugs or new type of infectious disease markers that you would like to go after. You need to be able to search that type of diversity. And so you really can't even do that within a microbial platform. You need to be doing that in vitro. And then after you've done some of your initial development and enrichment on the in vitro setting, you can take that and directly port it over into your yeast system. And we've actually shown that there is correlation between our in vitro measurements and our yeast measurements. And so what we sort of develop and screen for in vitro will translate into our yeast system. We can then do things like multicolor fluorescent activated cell sorting to begin to sort of further refine and optimize our designs for the in vivo setting within a microorganism like yeast, where we can screen on the order of millions of different variants. And then ultimately, we can take our best performing switches and begin to translate them into our human cells and begin to think about different applications, right? And certainly there are applications for these types of control systems in gene therapy, sort of vectors, right, where you want to control the therapeutic protein or the protein that's being produced in response to particular inputs. We've done a lot of work with sort of immunotherapy and immune system engineering, which I'll touch upon in the next set of slides. And there's also sort of applications for these types of control systems within sort of the stem cell space as well. Okay, so now I'm going to go into a particular application that we've been working on with a clinician who's up in Seattle at the Children's Research Hospital and Fred Hutch Institute. And this is on sort of developing basically better immune system based therapies. And so the basic idea is that in this type of immunotherapy application, you're going to harvest lymphocytes from a patient. You're going to basically recover the desired cell type and then begin to engineer that desired cell type with particular types of therapeutic functions. And this is a very active field of research. And there's basically a lot of different types of activities that people have looked at programming into these types of cells. And in particular, the ones that we've looked at with our collaborators are sort of chimeric antigen receptors to redirect the specificity of the T cell. Also, the expression of different types of cytokines so that you can self-sustain the cell within the human patient, and even different types of chimeric cytokine receptors. So these, and then after you do the engineering, you're going to transfer the engineered cell back into the patient, and that's going to become part of the therapy that's actually given to the patient. So what's the challenge? So as we said, this is a very active area of research. There are a lot of clinical trials that sort of go on in this field. And in particular, now a couple years ago, there was a lot of excitement around some successes that had come out of some early clinical trials with this work. And in particular, what they saw was that when they used this to treat CLL, that they saw complete and sustained remission after they had transferred T cells that were engineered with a CD19 CAR receptor. But what you also noted if you read the paper is that they encountered significant toxicities. And in particular, there's the chances, and what they observed was sort of cytokine storm and tumor lysis syndrome. And this is essentially because you have high-level activation of these CAR plus T cells. And what you also see is B-cell aplasia, where these T cells are basically recognizing CD19 plus B cells within the patient. So one of the reasons this comes about is because you don't, and the way that these systems are currently designed is you don't wrap control around the therapeutic activities that you've engineered into the T cell. Essentially, you have your CAR being expressed at very high levels, your chimeric cytokine receptor, and also different types of receptors for survival. And so you get these kind of off-target effects within the patient that can, again, result in significant toxicities. And so what you'd like to have is basically a control system that you can use to regulate the levels to which the different therapeutic activities are active, okay, and how long the therapy is going to be turned on within the patient. And so we believe that the RNA-based control systems that we develop can actually be very good sort of control systems to implement in this type of setting, and there's a number of reasons for that. But in particular, when you think about RNA as a regulatory molecule and you think about putting it within a human patient, it's sort of generally believed and been seen that for the most part, it's not going to induce an immune response, right? Whereas if we talk about putting sort of synthetic transcription factors or transcription factors that are taken from other organisms, then you have more of a concern that that's going to actually be recognized as non-self and induce an immune response. So these are very sort of clinically appropriate when you think about implementing a control system. And so we decided to do an initial study where we looked at how our RNA devices could be used to regulate chimeric cytokine receptors and cytokine expression to control T cell proliferation and survival as an initial test. And so this is essentially a schematic of the T cell proliferation pathway, and it's just to sort of indicate you have this architecture where you have an upstream signaling event, you have a signal amplification cascade, and then you have your pathway output. And so we decided that we would basically interface our synthetic control system with that upstream signaling event. And that was because we felt that we could develop very stringent switches so that we would have tight offs, right? And that we could take advantage of the signal amplification that would occur downstream of our control system to amplify the output of our system. And so again, the basic system design looks like this, where you have our ribozyme-based control system regulating a cytokine like IL-2. In the absence of drug, the ribozyme is active. You would get apoptosis or death. In the presence of drug, the ribozyme becomes inactive. Your cytokine is expressed. You should have proliferation and survival of the T cell. So again, in culture, we tested this in both mouse and human T cell lines. And basically, you can see that we have a fusion between eGFP and IL-2, so we can read out GFP expression and relative viability. And these, again, are in transient assays in culture. And so what you see is that when we have one copy of our system, there's a lot of leakiness. As we add 2, 3, and 4, we're able to systematically decrease that leakiness and then maintain the switching effect. And in particular, if you look at viability at the bottom, what you see here, this gray bar sort of indicates the basal viability in the absence of any transgenic IL-2 being present, so that's our sort of baseline. You can see that we get very close to that baseline, but we maintain very good switching activity. So we decided to go with these sort of 3 and 4 copy systems and integrate them stably within our T cells, and then look at the dynamic behavior of the system. And so at day 0 here is when we're adding our model drug, which in this case is theophylline. You can see the system responds within 1 or 2 days up to steady state level. When we remove drug, within 2 days it goes back down to the off state, and then again, if we add it again, it continues to respond. So this is just to say that you do have sort of dynamic response to the presence and absence of your drug molecule as it's being provided to the cells. And then the final thing we had done in this early system was essentially to look at whether this would translate to control within a mouse model. And so here we have T cells that are expressing luciferase, also expressing that construct that I showed you previously with our RNA device regulating the cytokine expression. The luciferase is just to image the proliferation of the T cells. This was a flank injection in the absence and presence of drug. And basically what you see is that these are the negative controls, the positive controls, and then our system where we've integrated the device. And you can see here we're getting increased proliferation in the presence of the drug and not in the absence. And after about 12 days, there's about a 20-fold change in the proliferation of the cells. So, right, so this was, sorry. This was to show sort of one example based upon controlling the proliferation and survival of the T cells. What we've gone on to do since that is a couple things. The first is that we've actually adapted the devices to respond to a pharmaceutical FDA-approved drug. So everything that I showed you previously was with a model drug, and once we showed that it worked and that the system could be sort of developed this way, we went in and sort of developed sensors that would respond to FDA-approved clinical drugs that you can inject into patients and give them as part of the treatment. And the other thing that we've done is we've developed this sort of dual system where you can have this RNA device regulating the transgenes such as CCRs and CARs, and then a microRNA or RNAi-based device which would regulate endogenous receptors like CD132. So you begin to sort of target multiple nodes within the network so that you can get increased sort of control over survival and proliferation of your T cells. And these are the systems that we're currently prototyping. And this is just showing you some of the early data that we have with the switches that respond to the pharmaceutical drug. Okay, and then I'm just going to quickly touch upon, in the last couple minutes, a sort of slightly different system that looks at how you can adapt these types of systems to regulate, to respond to endogenous markers to begin to build autonomous control systems. And in particular, this was a system that we built based upon alternative splicing. So think of it very similar to the ribozyme-based device, except that instead of a ribozyme, you now have a protein sensor, and your ribozyme is being replaced by this alternative exon inclusion module, with the basic idea that now you're regulating the inclusion of this middle exon. And the basic idea is that when that middle exon is included, it's going to give you low output of your protein because there's a stop codon that's integrated within it. And when it's excluded, you get high output. And so what we were able to show basically is that we could take this system, integrate aptamers for components within the NF-kappa B pathway, in this case either P50 or P65. And what you could see is that we developed a system that responded to P65. And also, if you look by fluorescence, in this case we're regulating GFP, you can see that when we induce signaling through the pathway, we get increased fluorescence. And when we don't induce signaling, the fluorescence is off. We can now, because we have this modular sort of capability, replace GFP with a suicide gene, in this case thymidine kinase, which is responsive to ganciclovir, a prodrug. And what we can show is that, again, in the presence of TNF-alpha and ganciclovir, we're inducing cell death. And you can see that also here as we quantify cell survival. And this is of our system as a function of ganciclovir. Similarly, because we have modularity, we can take that same sort of cell suicide system, replace the aptamer to NF-kappa B to an aptamer for a component in the Wnt signaling pathway beta-catenin, and we could show that it responds similarly in terms of inducing cell death when you induce signaling through the Wnt pathway and also have ganciclovir present. And again, you see the quantification here. Okay, so, and again, just to sort of highlight, yikes, sorry. What we're doing now with this type of system is expanding it to more complex types of alternative splicing patterns, sort of mutually exclusive alternative splicing patterns, with the idea that we want to use this not to just control expression of a protein, but we actually want to control, when you have modular exons, how they're being sort of combined together in a modular combinatorial type fashion, so you can start to reprogram proteins and not just control expression. With that, I will end and thank my laboratory, who's responsible for this work, and in particular, Kathy Wei and Yvonne Chin, who had done the work in translating the ribozyme-based system into mammalian cells, Yvonne Chen and Remus Wong, who have done the work on using the ribozyme-based system within T cells, with our collaborator, Mike Jensen, and then also Stephanie Kuller and Melina Mather for the alternative splicing work, and you for your time and attention.
Questions, please.
So is there a reason why you want to do the initial development with using a model drug rather than an FDA-approved drug?
Yeah, so the reason that, so the question is, why I think for the T cell project, did we go through the initial development with a model drug as opposed to like an FDA-approved drug? And the reason there was that, we had the sensor parts already generated for some of the model drugs, right? Ones that you wouldn't actually use in humans, but you could sort of do for your model studies. Whereas we had to go in and generate new sensors to the FDA-approved drugs. And we were doing that kind of simultaneously, right? So instead of sort of waiting to get the sensors, right, and then sort of put it in, because we had developed this in a modular platform, we could basically prototype and optimize and sort of understand, right, the sort of performance of the system, right, and sort of where you had to quantitatively tune things with the model drug. And then once we once we had the sensor to the drug that would actually be used in a clinical setting, we could swap that in, and we would anticipate, based upon other things that we've done in our laboratory, and also what we're seeing right now, that we would see similar behavior so that the optimization process would be faster, right? So we could get to the point where we're going into animal models basically faster with now the system that we would hope to use eventually in a clinical setting.
Also a question about the T cell work. So, I mean, we have demonstrated a kind of on and off controlling, either on and off. Can you envision that kind of what dosage control to kind of control the threshold? So maybe you spare some of the normal cells, but targeting cancer cells have a higher expression of the receptor you try to target.
Yeah. Again, so the question is, well, basically thinking about sort of the on-off, data that we show versus, the sort of intermediate ranges, right? And I think there's a lot of nuances there, right? So as I showed in some of the early data with the immortalized human cell lines, you could see that the system is titratable, right? And so as you have intermediate concentrations of drug, up to the high point, you're getting sort of titratable response in gene expression. So it's not sort of a digital response, right? And that's certainly true. And so you can imagine that that would sort of play a role. It becomes even more complicated, of course, when you're thinking about actually implementing this within humans, right? Because you have just the dynamics there of sort of how the drug is being injected into the patient, and then its sort of half-life, right? And then how it decays, right? So it's not the case that when you actually use it, there's going to be a constant level of drug that's going to always be present, right, within the human body. You're going to have sort of this response, right, of sort of going up to a high concentration, a half-life, and then it's going to be decaying, and then you'll get another injection, right? And so that is going to have to take some very careful studies, both within the animal model, and within sort of early clinical trials, if and when we get to that stage. But it does become quite complicated.
I had an additional question related to computational design. So the tools for predicting RNA structure are much better than that for protein structure, but the devices you're making, the RNA molecules, are quite complex. Are the computational predictors of RNA structure sufficiently advanced to be able to help?
So yeah, the question is sort of where are the computational tools for the type of design that we're doing? And what I would say is that, so there are some limitations to the computational tools that are currently out there, right? Number one, you're really just looking at secondary structure, right? And there are oftentimes tertiary interactions within the RNA that can actually play a role within the function, right? So for example, with the ribozymes, those two loops that you see off of them actually have a tertiary contact in order for it to be active, which is not captured necessarily in the model, so you have to take that into sort of consideration. The other thing is that most of the models, you're looking at sort of a minimal free energy, right? Which is not necessarily, what happens when you're in vivo, right? In vivo, you have a transcript that's being made. It could fold into sort of a local minima, but not necessarily a global minima. And then you can have kinetic traps because there's a time scale associated with it actually moving to, let's say, the global minima. And the RNA has a lifespan within the cell, so it might not actually be able to get into that global minima before it actually decays, right? So there are these sort of kinetic factors that come into play when you think about local versus global folding and the time scale that it takes for things to switch between different conformations, which are not really captured by the models at this stage. And so that being said, the models do help with some of the initial design, but you have to have these things in sort of consideration, things in mind when you're using them. And I think, as Jim sort of indicated before, I think it helps when you've done a bit of RNA design and you get a sense for basically how some of the things that you've designed based upon the models are working inside cells, and sort of what things are being captured and what aren't, and what things you kind of have to integrate into your own from your own knowledge. We are sort of working with another lab at Stanford to try to think about how we can develop better models, especially ones that would capture some of the things that you see with dynamic RNAs that switch between different conformations, because that's also not very well captured because of the kinetic aspect. So, but that's sort of where we are in the field right now.
Thank you very much, Christina.